Greetings, PsychU community. This is Rachel Self from Otsuka Neuroscience Medical Affairs. I'm joined by two of my outstanding colleagues, Drs. Heather Davidson and Carolyn Tyler. We will be covering the first in a series of slide decks for you, focusing on psychoneuroimmunology, an increasingly compelling topic in the fields of psychiatry and neuroscience. We'll begin with some basic information on the immune system and some of the key players in the inflammatory response. This is essentially to lay the foundation for future decks in the series that will take a deeper dive into the evidence and role for neuroinflammation in mood disorders and other psychiatric diagnoses. We have a lot of exciting ground to cover, so let's get started. This presentation is sponsored by Otsuka and Lundbeck, and some of the materials were developed in collaboration with Dr. Vladimir Malatak out of the University of South Carolina. As we dive into the discussion today, this gives you a little snapshot of some of our objectives. As I mentioned, we're going to provide an understanding of the immune system, some of the key cell types, how peripheral inflammation is transmitted to the central nervous system, discuss some of the key lines and processes underlying neuroinflammation, outline potential causes and consequences, and then talk about potential treatment modalities, including both pharmacological and non-pharmacological interventions that reduce inflammation. As we discuss the overview of the flow of the discussion, first we're going to cover basics of the immune system with defining exactly what inflammation is, differentiate between types of inflammation, peripheral versus central, talk about the inflammatory response within the brain, and review some of the underlying mechanisms involved, as well as the key cell lines and players. And then, of course, we're going to talk about potential causes from the environment and elsewhere of inflammation, as well as some treatment modalities. So let's get started with the review, a very brief overview on the basics of the immune system. The immune system has been referred to as the body's grandest sense organ. It's responsible for identifying what belongs to the body or self and then what is foreign. If something is determined to be foreign, the immune system then determines whether it's friend, as in the case of food, foe, as we might see with viruses, bacterial infections, toxins, etc., or neutral, as in some people's cases, pollen. The immune system is made up of various cells, proteins, key organs that all orchestrate responses to defend the body from invaders. These are widely distributed throughout the body, as you can see on the graphic, and you'll probably also notice that unlike some systems within the body, such as the circulatory system, which is a closed system, the immune system has many players that are spread far and wide with quite complex interactions that can be more easily described often with war or combat analogies. So as we look at the next graphic, we'll see how that plays a role a little bit more in line with the types of immunity that have been defined. So on the left side, you'll notice we have innate immunity. So this is considered our natural or non-specific immune responses. And it consists of physical barriers that can be thought of sort of as the walls and gates of the fortress of our body. So you think about tears, urine, skin, the GI tract, mucous membranes, hair, cilia. All of these are considered some of our physical barriers that function as part of the innate immune system, whose primary goal is to stop infection before it even has a chance to enter the body. You can see that it's further broken down into humoral and cellular aspects, and you can see some of the key components that fall in line there. And if you think a little bit more in terms of the war and combat analogy that I just gave us a few minutes ago, the innate immune system can be thought of as sort of the volunteer or citizen militia of your body's defense system, whereas when we talk about the adaptive immune system or adaptive immunity, which is shown on the right portion of the slide, this is our acquired or learned type of immune responses. So when we think in our war or combat analogies, this would be more in line with the elite special forces. This is something that has to be acquired over time, and it has to work in conjunction with initial exposure of the innate immune system to detect a threat. So keep this in mind. There are many different players when you think about humoral and cellular components of our various types of immunity. The elite special forces shown there are T cells and B cells that are part of that adaptive or acquired immunity. And keep in mind also that these two types of immunity, even though they're sort of segregated on this slide, they work together to recognize, neutralize, eliminate, and or metabolize invaders to the system. So it really is a joint team effort, much like the branches of our various military forces to defend our nation, or in this sense, our bodies, from foreign invaders. Now that we have an idea about some of the basics of the immune system, we're going to move on and actually define what inflammation is. So inflammation is generally considered a local accumulation of fluid, plasma proteins, white blood cells. It's really thought to be the cornerstone of pathology when we look at changes that are observed across our system that are signaling really injury or insult or potential disease to the host or to our body as we think in these lines. It's discussed in terms of acute inflammation as well as chronic inflammation. And the main goal really here is to tell the brain that the body has been infected, and it occurs as a result of exposure to harmful stimuli. This could be a number of different things, including microbial pathogens, irritants, or other types of toxic components that come from the environment. The main goal and function, again, is to get the correct fighters to the battle site. So we continue with our combat analogy. Acute inflammation occurs early, and it's usually very transient in its time frame. This is a very adaptive type of response, and if it is kept in check, then the body will resolve the inflammatory process and everything goes back to its normal homeostatic tone. Chronic inflammation occurs when we have persistent infection, and it can actually lead to things like autoimmune diseases. So essentially, when the inflammatory process outlives its normal protective role and it changes to an overly excessive extent in magnitude or duration, we can see various deleterious effects on the body, including different types of illnesses that are associated with a chronic inflammatory tone. These are often considered diseases of affluence in our modern society. We see these popping up more and more. And this is essentially just when that inflammatory response becomes a little too excessive in magnitude and duration, as we mentioned. And you can think about the things that we commonly deal with, such as obesity, cardiovascular disease, diabetes, dementia, cancer, and now growing evidence showing that many of our psychiatric illnesses have a chronic inflammation backbone. They're mediated by proteins called cytokines, or chemicals called cytokines, which we'll be hearing much more about in a few slides. And these really mediate immune cell recruitment and complex intracellular signaling that control mechanisms to characterize the inflammatory process or inflammation itself. The primary physical manifestations of inflammation are going to be things that we're familiar with: redness, swelling, pain, heat, loss of function at the affected area. And we now know that more than 2.5 times of the people living in our modern day society are dealing with some of these diseases of civilization that have this inflammatory component. When we think about the types of inflammation, you can really differentiate this based on the location of occurrence. So on this slide, we notice that there is peripheral inflammation, which is inflammation that occurs anywhere in the body outside of the central nervous system. Again, it is in response to an acute injury or insult. It's really designed to aid in repair, resolution, or restoration of normal functioning. And it can stimulate different types of behavioral changes within the brain, such as fever, decreased activity or appetite, changes in cognition and attention, as well as even anxiety and depression. A lot of these changes have become known as sickness behavior, which we will discuss a bit more as we get into our topic today. Central nervous system inflammation or neuroinflammation is inflammation that occurs within the brain and the central nervous system, and it is characterized by chronic glial activation, including microglia and astrocytes, and to a lesser extent, the oligodendrocytes. Again, we'll be hearing more about this in just a few minutes. And it's marked by an exaggerated expression of some pro-inflammatory mediators. So those cytokines that we saw on the previous slide, which include a multitude of different chemicals, including reactive oxygen species. There is still a debate whether neuroinflammation is a cause or consequence of neural dysfunction. As we transition into how the inflammatory response is transmitted into the brain, the graphic that will be depicted on this slide shows us at least 4 different mechanisms by which peripheral inflammation is known to be transmitted to the central nervous system. So starting on the left portion of the slide, you'll see a small box that has HPA-axis depicted. This is a contributing factor to perpetuate the inflammatory cascade. And we know that hormones affiliated with the HPA-axis can be critical mediators of continuing this inflammation story within the body, including within the brain. Moving over a little bit to the right, you'll see a black arrow which indicates pro-inflammatory cytokines. These themselves can signal and trigger that there's a peripheral inflammatory response mounting and that the brain may need to get involved. As we look towards the middle portion of the slide, right above the disrupted barrier red box, you'll see immune cells. So circulating immune cells themselves can transmit inflammation within the body and get the brain incorporated to increase inflammatory tone within the central nervous system. Finally, if you look just above that, you'll see the vagus nerve. So predominantly mediated through the autonomic nervous system and its innervation of the lymphatic system, the vagal nerve afferents will actually relay cytokine signals to relevant brain areas. And this is a primary way by which we know peripheral inflammation is transmitted to the brain. On the far right portion of this graphic, you'll notice that there's some information regarding neurotransmitter synthesis, which we'll be hearing more about in a few slides as well. But just keep in mind there are multiple different processes that can be involved in this. You'll also notice gut microbiota at the bottom of this graphic that we'll talk about towards the end of the presentation as well. Just keep in mind there are multiple different mechanisms. This graphic is just depicting quite eloquently at least 4 of which we know inflammation is transmitted to the central nervous system. So as we progress here in our discussion, the table shown on this slide is giving us a glimpse at some of the key players that underlie neuroinflammation. We're going to jump off talking about some of the glial cells and their various roles, as well as the cytokines, which I've already teased you with a little bit in the previous slide. And then I'm going to hand it over to my colleagues to discuss the roles of glutamate, mitochondria and oxidative stress, the HPA-axis, as well as some key compounds and components that are being appreciated more and more for their role in mediating inflammatory pathways. That's S100B and arachidonic acid. But first, let's talk about the glial cells. We mostly hear within psychiatry and neuroscience, at least historically, about neurons. But we are really not appreciating as much as we should, hopefully this is changing, the role of various glial cells. And I will argue as we get through the next couple slides that these are really the unsung heroes or superheroes of the central nervous system. They're not just glue, which is where their name derives from, that are holding the neurons in place. They actually outnumber neurons by about 10 to 1. So if we think that there are about 100 billion neurons within our brain, there are at least a trillion glial cells. The microglia are our primary immune cells within the brain. They are thought of as the sources of homegrown inflammation, or the macrophages of the brain. They are truly the bilingual glial cells, so they can live and converse just as easily with neural language as they can immune language. They change phenotype and produce soluble factors in response to cellular damage and pathogens, and they can produce both pro and anti-inflammatory mediators. The graphic shown here depicts multiple different forms of microglia that exist within our brain. So you can see surveillant microglia shown here. They're actually the scanners of the brain, so they scan our entire brain in a matter of hours to monitor function and state of all the other glial cells, as well as vessels, synapses, and neurons. You can also see the pruning microglia depicted on this slide just below the surveillant microglia. These are really important in the developing central nervous system, and they interact and collaborate with all other cells to modulate brain activity, especially in those developmental timeframes. Neuromodulatory microglia are another type of glial cells that you can see depicted on this graphic, as well as proliferating microglia, and then the phagocytic microglia shown at the bottom right portion. These are involved in direct removal of dead, damaged, or alive cells that need to be cleared from the circulating environment. So if you just keep in mind, microglia are the primary source of pro-inflammatory cytokines within the brain, but they also release neurotrophic factors, including nerve growth factor, brain-derived neurotrophic factor, and glia-derived neurotrophic factor. They are active sensors of disturbances within their microenvironment. They're key to determining the well-being of our central nervous system. The astrocytes, I'll try to highlight for just a moment here. They are actually our most abundant glial cell type. They are involved in modifying permeability of the blood-brain barrier, and they proliferate after inflammation to restore brain homeostasis. Their primary role is in maintaining synaptic integrity and really maintaining the peace and the balance of the brain. As much as 50% of our astrocytes are actually GABAergic cells within the brain, and they can morph and change their function with varying degrees of specialization and differentiation in response to physiological and pathological demands, including immune activation. Each human astrocyte actually contacts and encases about 2 million synapses, and their main roles here are to remove excess neurotransmitter, synchronize neuronal activity, and form what has been referred to as an astrocytic cradle to really maintain this homeostasis. They're also highly involved in glutamate modulation in various different aspects, which Heather's going to be telling you a little bit more about. Finally, the oligodendrocytes, which are personally my favorite of the glial cells, they work in concert with the astrocytes and other glial cells, and they're best known for their production of the myelin sheath. Each oligodendrocyte can actually produce myelin for up to 60 different axons. And this is critical in ensuring the speed and agility of neurotransmission. They can also produce neurotrophins that can affect microglial expression, so they can help in calming some of those activated microglia down if needed. They're involved in glutamate homeostasis and excitatory neurotransmission and immune regulation. They're really thought of in terms of our white matter proportion of the brain. So now we've talked a little bit about glial cells and some of their vast roles within the central nervous system. Let's shift over and discuss cytokines a little bit more. This is a really nice graphic that I will try to walk you through and give you a good depiction of exactly what a cytokine is. So cytokines are really chemicals that are produced during an immune response, It's a broad and loose term that's used for all of the different components that are shown in the center of this graphic in the black circle. So chemokines, different inflammatory mediators. If you think of interleukins, we've probably all heard a lot about IL-6, and if not, you're going to be hearing a lot more about it soon. Reactive nitrogen species and reactive oxygen species. They can exist in multiple different forms, ranging from highly pro-inflammatory to very potently anti-inflammatory. They are produced and released by glial cells, again, highlighting the importance of glial cells within maintaining balance and proper functioning of our central nervous system. And release of too many pro-inflammatory cytokines can actually result in excitotoxicity, mitochondrial dysfunction, and oxidative stress. Some of the components that Heather's going to be talking with you a little bit more about. They are shown to be correlated with specific changes in behavior that are also associated with many of our psychiatric illnesses, including mood disorders, such as altered sleep and cognition, food intake, hormone regulation, and even anxiety and depression. The graphic shown here is giving you an indication of how increased release of cytokines can engage the microglia, altering neurotransmitter expression and production, with serotonin being the example provided on this graphic, as well as lead to changes in the astrocytes. If microglia are activated for a prolonged period of time, they will engage the astrocytes, which can then withdraw some of their trophic support to the oligodendrocytes, shown at the bottom left of this graphic, which can then lead to pulling away of the myelin sheath from axons, which we know is important for the proper neurotransmission within the brain. Can also lead some changes with the glutamatergic system, which could result in excitotoxicity and even reduction of neurotrophic support downstream. We'll be hearing more about this as we move along. As we transition to the next slide, we'll see that cytokines have been classified along their spectrum in a couple of different ways. Cytokines were actually first described in 1957, and we know that there are at least 300 different kinds, not all of which obviously are shown on this table. I'm not going to be going through this table in any sort of detail, but I just want this for your reference so you can get an idea of the key cytokines that might be involved in our adaptive or acquired immunity, as well as some of the ones that are shown to have that potent pro-inflammatory aspect, as well as some that can be strongly anti-inflammatory. With that, I'm going to hand the presentation over to Heather.
Thank you, Rachel. That was a really great overview of the immune system, and really, I think, is going to make my job a lot easier as I go through, the different cell types that you've talked about and how they really produce changes in the brain that affect our behavior. So on the next slide, I'll walk you through a hypothesis, the neuroimmune network hypothesis, that was proposed by Nussleck and Miller, and it's based on observations from preclinical animal models, and really it's meant to provide a framework so that we can understand how sensitization of the immune system could shape behavior in a way that can further perpetuate neuroinflammation. So as shown here, they hypothesized that early life adversity, or early life stress as it reads on the slide, sensitizes the immune system and causes peripheral inflammation that is transmitted to the brain through activation of the microglia, and this leads to changes in connectivity in behavioral control networks, leading to maladaptive, risky behaviors that further perpetuate inflammation. So you can see that down in the bottom right-hand side of the slide. So next, let's drill down to the synapse to see exactly where that disconnection occurs and the role that inflammation plays in that process. So synaptic glutamate, so Rachel kind of touched on glutamate when she was going through some of the glial cells. Synaptic glutamate is primarily controlled by the glutamate transporter, which is expressed on glial cells. And here you see it on the right-hand side, you see them located on astrocytes. The role of the transporter is to control glutamate and keep it contained within the synapse. So it clears any excess glutamate from the synapse or from around the synapse. Inflammatory cytokines can directly impact glutamatergic signaling by decreasing expression of these transporters, thereby allowing glutamate to spill over outside of the synapse. And this can lead to loss of specificity over information flow and subsequent neurodegeneration, as we'll see depicted in the next slide. So on the left-hand side here, you can see that in early or transient inflammation, it activates microglia, as Rachel mentioned. You see the activated microglia there in orange. Once the microglia are activated, they begin to generate additional inflammatory cytokines and elevate extrasynaptic glutamate through the kynurenine pathway that Rachel mentioned earlier. So when the kynurenine pathway is changed, it really shifts the synthesis of kynurenine away from serotonin and towards potential excitotoxicity for the neurons. Activated microglia also cause an elevation of reactive oxygen species and reactive nitrogen species that are damaging to oligodendrocytes, Rachel's favorite glial cell, and astrocytes that are critical for maintaining connectivity across neurons. So we'll come back to this story on reactive oxygen and nitrogen species in just a moment. On the right-hand side of the slide, we see that as inflammation becomes chronic, you notice the difference in the morphology of the astrocytes. The astrocytes retract, and the astrocytic cradle that is around the synapse between two neurons is lost. And this allows glutamate, again, to spill over from the synapse. Extrasynaptic glutamate leads to shrinkage of postsynaptic dendritic spines, and that's what you see on the far right there, the green postsynaptic spine there. The spines will shrink and retract. In addition to that, the loss of oligodendrocytes results in decreased myelination of neurons, which slows the propagation of action potentials. So as Rachel mentioned, one oligodendrocyte can myelinate up to 60 neurons. So think about that, losing one, the impacts that would have on transmission of information through the brain. So together, the loss of myelination and the loss of dendritic spines is what really produces the disconnectivity that was proposed in the neuroimmune network hypothesis that I showed you a few slides ago. So in the next slide, we're going to drill down a bit further to the subcellular level. So we can drill down a bit further to the mitochondria within the glial cells and neurons. So the main role for mitochondria is to generate ATP, and they do this through a process called oxidative phosphorylation. So we need ATP to be able to have our cells to survive and do what they need to do. But a byproduct of that oxidative phosphorylation reaction, or OXPHOS, is reactive oxygen and nitrogen species, which are damaging to proteins. So the inflammatory cytokines that are generated when microglia are activated will interfere with mitochondrial function and result in generation of more of these reactive oxygen and nitrogen species. This will lead to excitotoxicity and apoptosis, and ultimately leading to neuronal death. So this becomes a cycle where reactive oxygen and nitrogen species cause activation of microglia, which then release additional inflammatory cytokines, which interfere with mitochondrial function. This causes apoptosis and neuronal death. The potential toxicity of reactive oxygen and nitrogen species in the brain is normally counteracted by antioxidants such as glutathione or N-acetylcysteine. But when excess reactive oxygen and nitrogen species are produced, oxidative or nitrosative stress can cause cellular injury and further increase inflammation. So you can see how this is really a positive feedback mechanism. So in the next slide, we'll kind of take a step back and look at another type of insult, which is, and I touched on this earlier, but early life stress. And I mentioned in the first slide I started on that early life stress primes the immune system components for a pro-inflammatory response. And we know that early life adversity is associated with higher incidence of psychiatric illness in adulthood. So another piece of this may be the dysregulation of the HPA-axis, as Rachel mentioned when we were just starting. The HPA-axis has a negative feedback mechanism in place where the glucocorticoid receptors can turn the response off. They can turn the stress response off. But with chronic or early life stress, the receptors become resistant and this negative feedback mechanism no longer works. And this results in reduced inflammatory control and chronic cortisol release. In the next slide, we'll talk about how this plays into major depressive disorder. So psychosocial stressors activate the same brain regions that have evolved to respond to physical danger. Dysregulation of the HPA-axis and inflammation that result from exposure to these stressors may contribute to the symptomatology that we see in major depressive disorder. And as Rachel alluded to earlier, some of these symptoms are also referred to as fitness behavior because many of the behavioral symptoms of physical illness and depression are similar, such as social withdrawal and increased need for sleep or sleep dysregulation and dysregulation of appetite. So in the next slide, we'll touch on two additional contributors to neuroinflammation. S100B is a protein that's exclusively expressed in the brain, and it has a lot of different roles, but it's involved in cell-to-cell communication. At low levels, or low dose, if you want to call it that, it serves an anti-inflammatory role. But at higher levels, it promotes inflammation. Plasma levels of S100B correlate with many psychiatric illnesses, and very preliminary studies suggest that derivatives of this protein may eventually or ultimately serve as biomarkers for specific psychiatric disorders. And the next is arachidonic acid. This is a precursor for prostaglandin synthesis, and it's important for downstream inflammatory effects such as fever and pain sensitivity. It's converted to prostaglandin by COX-1 and COX-2 enzymes, and animal studies have shown that COX inhibitors may improve depressive-like behaviors in certain rodent models. These data are compelling, but the human studies have not shown much success with using these inhibitors as monotherapy in mood disorders. So there's more to the story than that. So hopefully this was a good overview of how inflammation leads to changes in the brain and our behavior. And I'll turn it over now for a more in-depth look at the causes of inflammation.
Thank you, Heather. That was fantastic. Now we're going to talk just very briefly about some potential causes of inflammation, and then I'm going to hand the baton over to Carolyn so she can take us home with some potential interventions which might reduce the inflammatory tone. So as you can see on this transition slide, we're looking at some of the potential triggers that can be involved in causing inflammation. So inflammation can be caused by different types of factors that exist within the environment, whether that be allergens, things that we're exposed to from work, irritants, which could be changes in temperature even, cold air. Some of us are very familiar with that. Different types of strong odors. Interacting with individuals in the environment is probably our largest source of potential stress and inflammation. So stress and emotions are involved, as well as exposure to certain types of foods or food additives. There's a growing amount of literature showing that certain types of overly processed foods and additives can significantly increase our inflammatory tone and levels of dysbiosis within the gut, which hopefully we'll have a chance to get to in a little while. But if we drill down a little bit further and talk about different types of environmental contributors to neuroinflammation, these can be coming from sources either outside or inside the body. So when we think about inside the body, we think about genetics, which is a topic that we'll look at a little bit closer in just a moment. But in terms of the outside the body types of contributors, we think about chronic stress, different types of harsh family environments. Heather talked to you a little bit about that early life adversity or early life stress. We know that the developmental period is really critical in terms of timing of development within the overall body, particularly the brain, and can be a very vulnerable time frame. We know that these types of stress, early life adversity, etc., can activate the innate immune system, and this can lead to changes which will put the body in motion for more chronic, long-term immune changes and chronic inflammation. In terms of mitochondrial toxins, MPTP is a prodrug or neurotoxin that is known to cross the blood-brain barrier very easily, and then it can be converted to toxic forms which actually interact with glial cells and metabolism, changing mitochondria and killing dopaminergic cells, can lead to very rapid onset of some deleterious symptoms which mimic Parkinson's disease. And a lot of this literature comes from preclinical or animal models. However, we do see mitochondria conserved highly across species, so a lot of these changes will happen in humans as well. When we think about internal or microenvironment types of changes, you can see here there's reference to infections and peripheral immunity, which we've already been talking quite a bit about. So this is obviously transmitted to the central nervous system, that we know there are at least 4 different distinct mechanisms by which that can happen, that we discussed a little bit earlier. And then the gut microflora. So this is really a burgeoning area of research, and we are starting to appreciate more and more the huge role that the gut actually plays in the development of the immune system, as well as being a physical barrier to aid in that innate immunity prior to something really invading the body. We know that 70% of our immune cells actually lie in the gut and are produced and developed in the gut. And the amount and type of bugs that we have in our gut really determines our reaction to stress lifelong. There's a growing appreciation for folks who will develop a leaky gut and how this can throw off the balance of T cell differentiation and really our inflammatory process throughout our lifespan. Maternal inflammation is another internal or microenvironmental type of source that can lead to a pro-inflammatory component. Again, we have a lot of preclinical animal research looking at various types of maternal inflammation, as well as having some pretty strong clinical evidence, particularly in individuals who go on to develop schizophrenia and autism. There's quite a bit of evidence suggesting that various types of viral infections and maternal inflammation or infection during certain terms of pregnancy can actually lead to an increase in your propensity and vulnerability to develop certain types of psychiatric and neurodevelopmental disorders. And then, as we've discussed all along, there is also low levels of stimulation in the microenvironment that can keep microglia primed, as opposed to returning to their normal surveillance state where they're just kind of keeping everything in balance and making sure everything's working properly. So these types of changes that would turn microglia into that chronically activated state as opposed to a calm surveillance state can also lead to inflammatory changes. So as we transition, we'll talk just a moment about genetics. I'm not going to go into a whole lot of detail here. This graphic is just showing us that there can be obviously genetic predispositions for the development of various psychiatric illnesses, which we're familiar with, but there can also be genetic predispositions that will alter the way that we respond to stressors in our environment or potential threats to our immunity. The growing appreciation now is that epigenetics probably is playing a larger role when we think in terms of psychiatric illnesses. And this can lead to changes of what is expressed or what is not expressed at the level of our genetic code. This can lead to changes in plasticity as well, which can cause a lot of problems long term if you are overexpressing things that could be deleterious and maybe underexpressing healthy aspects that could potentially lead to resilience if you are exposed to extreme stressors. So with that, I'm going to turn it over to Carolyn to talk about some potential anti-inflammatory treatment approaches.
Thanks, Rachel and Heather, for that fantastic overview of the science of psychoneuroimmunology. And given those factors, a variety of potential anti-inflammatory treatment approaches have been proposed and studied for psychiatric illnesses. So let's examine those further. So pharmacological approaches can directly or indirectly target the underlying causes of neuroinflammation. Direct inhibitors of inflammatory cytokines and their effectors are currently being studied. While these molecules may not enter the CNS, the peripheral administration of these molecules has shown some efficacy in certain models of depression. Reductions in oxidative stress could be an effective anti-inflammatory treatment strategy as well, as Heather discussed earlier, the effects on mitochondria and the role of oxidative stress in neuroinflammation. Steroids can directly inhibit immune system activation and inflammation, and then neurosteroids are actually being proposed as a new class of molecules to treat psychiatric illnesses like depression. NSAIDs, as Heather discussed earlier, are COX-2 inhibitors, and they have been observed to have variable efficacy for a variety of psychiatric illnesses, including depression, bipolar disorder, schizophrenia, and obsessive compulsive disorder. But further research into these molecules is required to understand their mechanism of action and their potential use in these disorders. Some antibiotics are known to readily cross the blood-brain barrier and enter the central nervous system. And in some studies, they have shown antidepressant effects. These studies are also ongoing. A variety of non-pharmacologic anti-inflammatory treatment options have been suggested and researched. These include caloric restriction and intermittent fasting, cognitive behavioral therapy, enhanced nutrition, exercise, increased socialization, meditation and mindfulness, probiotics, prebiotics, and symbiotics, and sleep. Further research is required to determine the efficacy of these non-pharmacologic treatments and their synergy with pharmacologic interventions for treating psychiatric disorders. In summary, the human immune system is a highly evolved and complex structure, and we know that inflammation underlies many diseases and disorders of modern society, impacting both the peripheral and central nervous systems. Multiple components of inflammation have been elucidated, but we still have a lot to learn, and there's quite a bit of ongoing research in this field. Multiple intracellular and environmental sources may contribute to neuroinflammation, and a growing body of evidence suggests neuroinflammation contributes to pathology of certain psychiatric disorders. Various pharmacologic and non-pharmacologic strategies can target and reduce inflammation, and again, there's ongoing research into this, so we expect more on the horizon. Thank you for joining us today for the basics of psychoneuroimmunology. For further information on this topic, please view part 2 of the series, The Psychoneuroimmunology of Mood Disorders.